Для церемонии открытия 21-го Менделеевского съезда на сцену приглашается президент Российского химического общества, академик Аслан Юсупович Цивадзе.
Двадцать первый Менделеевский съезд по общей и прикладной химии объявляется открытым. Уважаемые участники съезда, мы начинаем пленарное заседание. В соответствии с программой для пленарного доклада на сцену приглашается лауреат Нобелевской премии Жан-Пьер Соваж. Жан-Пьер Соваж, профессор университета Страсбурга, французский исследователь в области координационной и супрамолекулярной химии, а также химической технологии. В 1971 году он защитил диссертацию, посвященную синтезу и исследованию криптандов, выполненную под руководством лауреата Нобелевской премии Жан-Мари Лена в университете Луи Пастера в Страсбурге. В 1973-1974 годах под руководством профессора Малкольма Грина в Оксфорде он начал исследования в области химии переходных металлов и металлоорганической химии. Затем, после возвращения в Страсбург, в лаборатории Жан-Мари Лена он начал исследования в области искусственного фотосинтеза, фоторазложения воды и электролиза углекислого газа как новых подходов к созданию альтернативных источников энергии. В 1983 году в группе под руководством Жан-Пьера Соважа вышла первая статья, посвященная синтезу катенанов, и положившая начало активному развитию новой области химической науки, химической технологии, что в итоге привело Жан-Пьера Соважа к получению Нобелевской премии по химии в 2016 году совместно с сэром Фрейзером Стоддартом и Бернардом Ферингой за создание молекулярных машин. Жан-Пьер Соваж является автором более 500 публикаций, и согласно сайту Web of Sciences, он является одним из наиболее цитируемых авторов. Жан-Пьер Соваж является лауреатом множества премий различных химических сообществ и профессором ряда ведущих университетов мира. В 2016 году он стал иностранным членом Российской Академии наук. Жан-Пьер Соваж.
Thank you very much, Professor Aslan Civadze, for your introduction. Is there a laser? The laser doesn't seem to work too well. Was it? Okay. So let me first thank the organizers of this fantastic meeting, and Professor Civadze, and all the people who contributed to organize the meeting, the organizing committee. I'm, of course, very happy, and I feel very honored to be present, to participate in this great meeting. Just a word also, let me thank some of my Russian friends, Professor Yulia Gorbunova and Dr. Alexander Martinov, for their constant friendship and for helping me, generally speaking, in Russia, but also for the very pleasant contacts we have had for 15 years or so. So let me start my scientific lecture. I try to be very well organized, and I have a timer. So we'll talk about catenanes, rotaxanes, and molecular machines. And this is more or less the topic of the Nobel Prize. A statement, very simple statement. So how did we start? Why some people became interested in molecular machines? This is more or less explained here. If you take synthetic molecules, very generally speaking, these molecules are considered as static objects. They don't move, or if they move, it is completely at random. There is no control over their motion, and this has been a constant feature of synthetic molecules. Now, looking at biology, if you look at biological systems, it is totally different. Controlled molecular motions in biology are absolutely essential, and they are found everywhere. Any important process involves large amplitude motion in a strictly controlled fashion. So there are, for instance, rotary motors, linear motors, molecules which walk, walkers, molecular systems which contract or elongate. So these systems belong, for most of them, to a very wide family of proteins, the motor proteins. Motor proteins are everywhere in biology. Just two examples, very famous examples, which probably many of you already know. ATP synthase, it's a rotary motor. It is, of course, an extremely important enzyme because it is responsible for the synthesis of ATP from ADP and inorganic phosphate. It is also responsible for the hydrolysis of ATP to ADP and inorganic phosphate. It's a fantastic rotary motor which is present in any living organism. Another example, which is really spectacular, is that of kinesin. And kinesin is a walker, as I already mentioned. It walks on microtubules, mostly to transport matter from one spot in the cell to another spot where it is needed. A very beautiful video, which you can easily find on the web. This is that of ATP synthase. I should say that it's not science fiction. It is based on very precise scientific data. So ATP synthase is located on a membrane in the mitochondria. And there is this part at the bottom which spins. There is an axis here which is threaded in the hollow part of the big cluster of proteins, red and green here. And as it spins, ATP is synthesized from ADP and inorganic phosphate. It is very slow on the slide, but if you look at the real system, the real stuff, it spins very, very fast. a few thousand times per minute. Now, we will change completely topic and talk about catenanes and rotaxanes. Catenanes are interlocking rings, and the 2-catenane, which is drawn here, is the simplest member of the family because it consists of 2 interlocking rings. So 2 holds for the 2 rings which are interlocking with one another. Rotaxanes are somewhat similar. A rotaxane consists, or the simplest rotaxane, consists of a ring threaded by an axis, and at the 2 ends of the axis, we will attach big stoppers to prevent the system from dissociating. Why are catenanes and rotaxanes so exciting in relation to molecular machines and molecular motors? Very simple. If you take a very simple rotaxane, like the one here on the left, if you are able to move the ring from the left position here to this position on the right and go back to the left, you are very close to making a linear motor, very similar to a piston moving in a cylinder. And this, of course, will lead to a very simple molecular machine. But nobody had done that before the work of the various people working in this field of molecular machines. Similarly, if you have a rotaxane of this type, and if you can rotate the ring around the axis, or here, rotate the red ring within the blue ring, which is considered as fixed, you are on your way towards rotary motors. So you can pirouette, or you can even rotate. So this is why catenanes and rotaxanes are so interesting in relation to molecular machines. And basically, catenanes and rotaxanes led to this new field of molecular machines. Now let's try to make catenanes and rotaxanes. Not so simple. Chemists have been talking about catenanes and rotaxanes for more than a century. So this is a very old concept. But experimentally, the development of this field has been very, very limited. And I just would like to refer to some historical work done by Professor Schill in Freiburg in Germany. It's a neighbor city, very, very close to Strasbourg, my own city. And in 1964, so already many years ago, Schill and Lüttinghaus published a beautiful piece of work on the synthesis of a catenane. So that was the first time that a catenane was made convincingly. It was a great publication, very, very elegant. But the problem, or the weak point, let's say, was that it was a 22-step synthesis, so exceedingly difficult, and the overall yield was very, very small, very tiny. So chemists were really very impressed by the work, but nobody really invested work and time in this field because it seemed to be so difficult to make such molecules that everybody was kind of discouraged. So something new had to happen. And I will show you how we started in this field. So when I started my group, after working mostly with Jean-Marie Lane, my boss, I was very much interested in photochemistry, and in particular, there was a big project. Many groups were working on this project, which was trying to split the water molecule to H2 and O2. A big project, of course, very, very ambitious, because it's a dream reaction. And at the time, there was a hero in the 70s, in the 80s, in the 90s. It is still kind of the hero in the field. This is ruthenium traced by pyridine. Just to remind you, in the Mendeleev classification, ruthenium is just below iron. Ruthenium is a noble metal, which means that it is very, very expensive. And our project was trying to replace ruthenium by another metal, much, much cheaper. And so we embarked in a project aiming at using copper instead of ruthenium to make very interesting molecules in relation to photochemistry and electron transfer. So the work started in collaboration with a team from America, David McMillan. I'm very sorry. I don't know if somebody has some water. So we started a project on copper complexes with David McMillan, as I said. And David McMillan spent a year in Strasbourg. He was on sabbatical leave. And we had made the molecule which is represented here, a very simple molecule. Dave McMillan was more of a physicist than a chemist. He was a photophysicist. And he was very excited when he saw the molecule. And he said, we must collaborate. And we started a collaboration immediately. So we started from this molecule, very simple molecule. We mixed with copper 1+, copper tetrakis acetonitrile, and very, very easily we obtained a copper complex. This molecule on the right, I don't know if you see my laser, but I don't think you see anything, do you? Yeah? Personally, I do not see it. So the copper complex, which is here, was very exciting in terms of photochemistry, but there was something else. And now we will try to discover the something else together. If you imagine that you link the point, very difficult, a point which is above here to another point, let me try. So the idea is quite simple, but it's very difficult to explain without a laser. You can make a ring in a vertical plane. You can make another ring in a horizontal plane. And those two rings will be interlocking with one another. Yeah? Microphone, yes, sorry. So you can make a ring in a vertical plane, another ring in a horizontal plane, and those two rings will be interlocking with one another. So you will see immediately how it works. So we had to decide, shall we be photochemists and stay photochemists or photophysicists, or move to another field, and we decided to jump to another field, which was that of catenanes and rotaxanes. Within a couple of months, we had our first publication, thanks to the talent of Christian Dietrich Büchecker, a great organic chemist, great synthetic chemist. And we published our first paper in 1983. Just a couple of months of work. It was published in French. Be careful, it's published in French. In a reasonably good journal, but not a high-impact journal. But the main reason is that high-impact journals didn't mean anything to us at the time. So this is a video which shows you how it works. So we start from the molecule, which is here, and we will mix it with copper, and we obtain some kind of N-twine complex. We make a ring on the right, another ring on the left, and here we are Thank you. Thank you very much. Thank you. And here we are. We have our catenary. And so we developed another strategy, which is slightly more complex. We made a ring, which is here. And now we will proceed to threading of the ring by another fragment, organic fragment, thanks to the effect of copper. Copper arrives, it forces the second fragment to thread in, and then we cyclize. So that was the very beginning of catenanes. I should say that all of a sudden, catenanes appeared as very accessible molecules. It was a three-step synthesis. The yield was really good. And we could make such molecules at the gram scale. So here are structures. This is the copper-containing species. We can easily remove the copper, and we obtain the method-free species, which shows that the system has completely rearranged. I should say in passing that we have been very much interested in molecular topology for years and years, and we have probably devoted as much effort in the field of topology as in the field of molecular machines. And we could make quite exotic topologies, very complex topologies, such as the trefoil knot, which is on the left here, or Solomon, the Solomon link, which is on the right, and other more complex topologies. And these molecules have really fantastic properties. But it's not the subject of today's lecture. Just to show you that knots and catenanes are everywhere, in DNA, in proteins, in viruses. And so we thought it was also a motivation for us to show that chemists can make complex topologies in a way not really similar, but reminiscent of what nature can do, knots with DNA, knots with proteins and viruses. On proteins, just one point. If you look at the crystal data bank in Cambridge, 1 and 2% of the proteins which have been crystallographically fully characterized are knotted. So knots are everywhere in any living organism. Now let's look at the first molecular machine we made in Strasbourg. It is built on a catenary. So this is very simple coordination chemistry now. We will start from here, on the left, the upper line, and we have copper 1 as the metal. Copper 1 plus is not really a transition metal. It's a D10 electronic configuration metal, and it is very happy if it is surrounded by 4 nitrogen atoms, as indicated here. And now we will oxidize, abstract an electron from copper 1 to generate copper 2. This is on the right part of the upper line. Copper 2 plus is now a real transition metal, and it wants to be 5 coordinated. So the ring, which is here, will move, will rotate, and the 3 nitrogen atoms, which were originally at the periphery, will replace the 2 nitrogen atoms originally interacting with copper, so as to generate the new species, which is on the right at the bottom line. So this is a half turn. We can go back, we reduce copper 2 to copper 1, we generate a very unstable species, which will rearrange. Again, the ring will glide, and we will regenerate the starting form of the molecule. So that was really the beginning, and it works beautifully. It's very, very clean. We never detected any degradation, any fatigue of the system. And just a small video to show you the principle. We go from copper 1 to copper 2. The copper-2 species is now very happy when it is 5 coordinated. We can again re-inject an electron. It will go back to the 4 coordinate, etc. So this is something which was really at the beginning of this field. To show you that although at the beginning it was very, very slow, we needed seconds to minutes to set the system in motion. But after 10 or 12 years of work, we could prepare very fast-moving systems, rearranging in the microsecond to millisecond time scale. At the same time, exactly at the same time, Professor Stoddart and some of his colleagues made a molecular shuttle, which is kind of a rotaxane again. It can move, as I said before, as a linear motor. So the ring can go from the green station to the red station and go back to the green station. And again, that was the beginning of the field on molecular machines, mostly in Professor Stoddart's group and some other groups. Now, what we have been doing, I'm not going to discuss many systems, but one, which is that of a muscle. Well, of course, it's not a real muscle. This is a bit abusive. But it's a molecular system which can contract and elongate at will. It is a slightly more complex rotaxane. It's a rotaxane dimer. So you have a rotaxane on the top, another rotaxane below, and we have interlinked those two rotaxanes, interconnected those two rotaxanes, so as to obtain a rotaxane dimer. And now we send a chemical signal to the molecule, and the molecule will contract. We go from 8 nanometers to 6 nanometers. We can elongate again, contract again, etc. And this piece of work has inspired many groups who have now developed materials, fibers, which can contract and elongate, and really very exciting systems, more towards applications than the original work we had done at the beginning of the years 2000. Just to tell you that catenanes and rotaxanes are not the only molecules which can be used for making molecular machines. And here are some very important names of people who've either used catenanes and rotaxanes or non-interlocking systems. And I will just show you the work of Ben Feringa and his group at the University of Groningen in the Netherlands. Ben Feringa was working on switches, molecules which you can convert from one form to another form. And they were working on photochemistry of double bonds. So if you have a CC double bond, when you shine light on it, you can isomerize it. And you can isomerize it to another species, an isomer. And they found, more or less by accident, that if you isolate the species which is on the right part, on the upper line, after maybe one night, it rearranges to another species, which is at the bottom now, on the right part. So the system has continued to move. And if you shine light, if you excite photochemically, you will have some motion. You will continue the rotation motion. And finally, by hitting, you go back to the starting point. And they published a beautiful video, which I hope will help you to visualize Feringa's chemistry, Feringa's system. So there are 4 steps. A thermal step, a photochemical step, a second thermal step, and a second photochemical step. So now we will start with light. And you see it starts, now you heat, you shine light, you heat, you shine light. And I think the video should show you that it rotates, that it's a one-direction motion. It's a real rotary motor. It's not pirouetting simply. It's a real rotary motor. So can some of you visualize the motion? I hope yes. I'm very sorry if you cannot. I cannot help you. So let me finish up with one of the most complex molecular systems we've made in our group, which is a molecular compressor. So this molecular compressor is a 4-rotaxane. 4, because now the molecule contains 4 molecular fragments, 4 components, which are not covalently speaking interconnected. So there is no covalent bond between any of those 4 components. So this is why it is called a 4-rotaxane. So we have 2 axes at the top and at the bottom. Some people call them dumbbells. And the middle here. Yeah, sorry. Yeah, you're right, thank you. And now, if you look at the center, you have a bis macrocycle on the right and a bis macrocycle on the left. And in between the 2 rings of each bis macrocycle, you have a plate. The green species is a plate. And the idea, the project, was to check whether we should be able to move the 2 plates and compress something which would be trapped between those 2 plates. So you have G, which is a guest, complex between the 2 green plates, and we will try to find some trick to compress the guest and modify its shape and finally expel it. So this is the project. So now let's look at the molecule. So this is the molecule we wanted to make. This is nothing for the moment but a research project. So we had long discussions in my group. Should we start the project? Should we try to make such a molecule? Because you can easily figure out that it's not a simple project. So we started. You see again, you have the 2 dumbbells, the 2 bis macrocycles, and the 2 plates, which are porphyrins, zinc porphyrins. So we succeeded, and it was much easier than expected because of symmetry and because of the magic template effect of copper. So on top of that, the cherry on the cake, we could even crystallize the molecule and obtain an X-ray structure, and this is the X-ray structure of the species. So it's a relatively heavy molecule, molecular weight of 9,000. And you see the 2 plates. So the plates are those porphyrins at the very center of the molecule. And in between those 2 plates, we can insert molecules, smaller molecules, which can be trapped in between those 2 porphyrins. So till now, the project works. Now, let's try to compress. And this is the trick. So now we start again from the X-ray structure. This is nothing but the X-ray structure. And the most important points here are the 4 copper ions, which are orange or golden. And if we remove those 4 copper ions, the system will be completely rearranged, will be completely modified, and this will lead to compression of the gas which is in between the porphyrids. So let's do that. Again, this is the X-ray structure. And now we send a signal. We remove the copper, and the system changes completely. One fragment will try to interact with a porphyrin, the second one with another porphyrin, and the space in between the two porphyrins is really very, very strongly decreased, so that the gas, which was originally complex between the two porphyrins, will be compressed, 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 and finally expelled. If we add copper again, so this is the other chemical signal, we regenerate the starting molecule with a relatively large cavity which can be occupied again by some small organic gas. So let me conclude on this part by saying that before the emergence of molecular machines, synthetic molecules were mostly considered as motionless objects or species undergoing stochastic motion. Molecular systems undergoing large amplitude motions in a controlled fashion were very, very scarce, almost unknown. And so this is, I believe, the main contribution that the various groups which worked in the field of molecular machines brought to the scientific community. Now, just a few minutes on applications or potential applications of this field of molecular machines and muscles. For the moment, it is still difficult to know exactly in which field these molecules will find important applications. But there are already answers, or let's say beginnings of answers. One commercial application, so it's really commercially important, is that of polyrotaxanes. So if you take large rings, such as cyclodextrins, which are polysugar, cyclic polysugars, and if you thread them with various very long chains, you obtain systems which are very schematically represented like that. And by connecting two rings, you have some slide ring function. And so you incorporate slide ring elements, and this is called the pulley effect, like a pulley. And the molecule, the organic backbone, becomes incredibly flexible. You have a gel, but it's a gel which is incredibly mobile in terms of molecular mobility. And so the materials are unique. They can move, as I said, and if you want to scratch them, you can scratch them, but very rapidly the scratch will be erased, and you will regenerate the initial form of the material. So you repair, or it is a self-repair process within a few seconds. And so this has been used for protecting screens of various displays and mobile phones. Some of them are still protected using this kind of gel as a protective screen. Now, let's be perhaps more futuristic. Professor Stoddart and his group have done beautiful work in collaboration with Jim Heath on storing and processing information with molecules. So this could be perhaps an interesting extension. Synthetic enzymes will also be certainly the subject of intense research, because enzymes are, generally speaking, kind of molecular machines. Molecules or molecular systems which can sort and transport molecules, in particular through a membrane. Now this is more futuristic, science fiction, right? But robots, micro-robots, nano-robots will need artificial muscles or will need other artificial molecular machines. Vectorization of biologically active species, so this is targeting, will probably be one of the main fields of application in the future. Long-term future or not, we don't know. But targeting tumoral cells is a very active field of research for the moment. And let me finish up by saying that in spite of the many possibilities offered by molecular machines in terms of potential applications for the future, one should keep in mind that basic research is still of utmost importance and is or has been at the origin of most of the technologies we are nowadays using in our daily life. Let me finish up by thanking some institutions, my university, the European Commission, which is, of course, highly supportive of research. And after the discussion we had before the opening ceremony, I wish the very best to the Russian Federation for having more contact with the European Commission and more contact with European scientists to establish networks and to collaborate. A big thank you to the people in my group, PhD students, postdocs, and professional researchers. And this is one of the specificities of the French system. We work with professional researchers belonging to the CNRS, the research agency, or members of the university. And so when I started my group, we were something like 4 or 5 professional researchers, very good friends, and we were working together. This can be extremely efficient. I had 2 great teachers. I'd like to thank my family and my 2 friends, Fraser Stoddard and Ben Feringa. And I think I will stop at this stage and thank you for your attention. Thank you.